right, as I'm getting this stuff loaded up, if you're tuning in, please mute. Me too. That always scares me when I hear myself start talking.
I'll get that. There you go.
Okay, we'll get started. Good morning, everybody. I see the transcript is working just fine. Holy cow. Any questions or concerns before I get going with stuff? The same is true for people tuning in on Zoom. If you've got anything, unmute and let me know. I'll give you a second. Hopefully everybody has had an opportunity to get logged into Hackback and started to get that stuff figured out. I have not clicked back on it since right before class started this week. But I'm going to do that after lecture today just to kind of see what's happening and what's really going on. Any questions or concerns about that? Okay. So to get started today, the first bit of this is really kind of introductory material, a little bit of historical stuff to kind of get us thinking about microbiology in the first place, right? I know you all have some ideas as you're coming in here and potentially things that I maybe biased you with, thinking disease is one of my favorite things to talk about. Maybe that's what you start thinking about as we start talking microbes, right? But there's a lot more than just that. And we kind of hinted at that the littlest bit Monday. So there are all sorts of potentials when we think about microbiology and some of the fields that you might potentially find yourself in. I said as we started this that my gut tells me most of you are thinking some sort of health professions. You kind of already know where your trajectory is headed, but others maybe not quite certain about that, and possibly you might find different ideas as we start thinking about this that might actually be something that you truly would like to dive a little deeper into. So in this instance, we're looking at oil spill a little over a decade ago, which is crazy to think that it was that long ago. The Deepwater Horizon oil spill in the Gulf spewed out massive quantities of oil and in turn caused all sorts of issues, decimating wildlife, fisheries, as we see this turtle covered in oil. Down, we probably could pan down into the boat and see the bottom to wash them off with. We should be doing some commercials right as we move through here. It's not what we're really thinking about. There are all sorts of other possibilities and things that we may recognize in the environment and then be able to tap into that and potentially utilize that for our benefits in other circumstances. So really, as we're looking at this, after the oil spill, they recognized some bacteria that are common, naturally occurring, but not thinking mass quantities of this stuff, because we need to think about their possible resources and what access do they have, right? Given the proper circumstances, the right situation, that sets the stage maybe for mass quantities of these organisms to flourish and to thrive in this, I guess maybe we could say, new habitat, right? So this bacteria, it's an ocean-dwelling bacterium, Alcanoborax. These guys, Borcumensis, I believe is the species. They're naturally occurring, but
Do I send a PowerPoint?
Excuse me?
How do I send a PowerPoint?
How do you send a PowerPoint?
Sorry, I was not trying to talk in that.
Okay. These guys, as the oil is spewing out, now all of a sudden, it's like this, small population, this few, were all of a sudden taken out to the buffet. They've got mass quantities of resources where maybe it was, desert-like before that, where they were just eking out an existence. And now all of a sudden, they're bathed in nutrients. And these populations flourish. We see them surging as they've got stuff to feed upon. We think about bioremediation like this, where we can take some sort of organism, possibly a microbe, something that we can utilize to help deal with specific issues. And those issues oftentimes are caused by us in the first place, and we try to mop it up, all right? So there's all sorts of ideas, more than just this, when we think about possibilities to utilize some sort of microbe to our advantage, all right? And it could be something like bioremediation, cleaning up all sorts of things, toxic waste or other stuff are possibilities. Scientists at the INL do research trying to figure out different types of microbes that can be useful in bioremediation of stuff like nuclear waste, right? And cleaning up some of these things, or heavy metals, and trying to basically use the bacteria to breathe, to use these molecules as resources, and basically take something that's possibly hazardous or is hazardous and make it more innocuous or inert, something that's not a potential hazard to us, all right? So there's all sorts of ideas. And across the way in biology, there are microbiologists that are thinking disease stuff. And there are others that are more along this realm, where they're thinking about potentials with microbes to do other things to benefit humanity. We could also switch it over and think about like food production, whether it's utilizing microbes to make stuff or ensuring that the stuff we are producing isn't tainted or contaminated with microbes. There's all sorts of possibilities, right? So we'll get more into some of those tidbits here in just a second. So as we think a little bit medical standpoint, sometimes a patient may present and maybe we're thinking some sort of infection, maybe meningitis could be a possibility, swelling of the lining around the brain, spinal taps, getting some cerebral spinal fluid lends to the potential to evaluate that and see maybe organisms are present or not. This stuff should be clear, like what we're looking at in our tube up here. If it's cloudy or turbid, maybe a little opaque, that's indicative of some sort of pathogen, most likely a bacterium, that's causing issues. There are numerous things in there besides just bacteria that causes the cloudiness, lots of white blood cells, and we'll hit details with that stuff and our mechanisms to try to deal with it in a little bit, all right? We're not worrying about it right now. So I was talking some food production, and Saccharomyces is a kind of yeast. Saccharomyces cerevisiae is a type of yeast that is used in food and alcohol production. And we have used this for a super long time, all right? Thousands of years. We have tapped into this microbe and used it for these benefits, all right? There's all sorts of things we think about with yeast cells. Fermentation is definitely something that they are very proficient at, right? We will think a little bit later about some of the ways organisms utilize resources, but yeasts are pretty cool, all right? These microscopic cells, these are all individual yeast cells. We've got a couple of them right here that are in the process of dividing. They replicate asexually. A single cell buds off a new daughter cell. We would call it a mother and daughter. And you get a genetically identical yeast cell, right? Bacteria kind of do things similarly. It's not exactly the same, right? The yeast cells are eukaryotic. They're like our own cells. There's membrane-bound organelles and nuclei. They're monsters compared to bacteria, right? Even though they're still microscopic, right? So in this instance, we can see a couple of circumstances. We've got some fermentation, we've got some flour, potentially some sourdough with, you know, all of the pandemic stuff, bread making and sourdough making, all of that stuff skyrockets. Everybody's like, oh, what am I going to do? I'll make some bread, all right? Yes, sir.
So if that yeast makes ethanol, why don't we get intoxicated when we eat bread?
That is a great question. Why don't you get intoxicated when you eat bread? Because in this process, these yeasts are feeding on the flour, right? They're pulling in the glucose molecules and they're metabolizing them. They're breaking them down just like our own cells would be if we're pulling in sugars, right? And as they metabolize, they release lots of carbon dioxide, the bubbles. That's what makes the dough rise. That's what makes your bread, the fluffiness, are these yeast cells that are doing that. And they're producing lots of carbon dioxide, right? If we took this little tub of yeasts and flours that are all, and there's sugar in there too, I'm sure a bit of that. As they're in there feeding on this, if we were to take it and seal it up so that there's no more oxygen that's available to these yeast cells, they still want to perform, they want to feed, right? But they don't do it quite as well. And that's when we turn into the fermentation pathways. That's when we start producing alcohol, right? So just making bread, they're just making carbon dioxide. But if you starve them of oxygen, then they would start producing alcohols as well. And I don't think your bread would turn out very well in those circumstances. But I don't know, right? It's worth the test, right? So there are definitely some potentials as this process is occurring for maybe, you know, minuscule amounts of alcohol are being produced in this process of fermentation, just in this vat or little jar of yeast and nutrients, right? And I would imagine maybe down towards the bottom of that, there's less oxygen available. Some of them ferment a little bit more. Once that starts getting baked up, if there was any minuscule amounts of alcohol that were generated, it would all evaporate in the oven. So you wouldn't even recognize it as happening. Unfortunately, or maybe fortunately, right? So if we shifted over and got rid of the bread and showed you some frothy beverages, beer or other substances the situation is pretty similar. And you would take whatever substance, if we were going to talk about the production of beer, you would have the malt, the barley or the wheat, the sugars within that would be stimulated with some enzymes to start breaking down so that we've got the monomers that are accessible to these yeast cells, not great big polysaccharides, right? We start breaking them down. Amylase is a great enzyme to do that. As that starts to get broken down into the malt, the yeast gets added to that. They start feeding. They start producing lots of bubbles, just like we see up there. But this wouldn't be in an open container. It would be in something that you can cap up. And once it has produced good amounts of bubbles, it's populated this big vat with lots of new yeast cells. Then we starve it of oxygen. We put the cap on it and it starts producing alcohol. And after you have done that for a period of time and you go to bottle it up, you can add a little bit of sugar or malt back to that, cap it, and in the bottle, it reacts and produces some bubbles. And that's how your frothy beverages get frothy in the first place. They carbonate themselves, all right? That's the idea, right? If you were making wine, it's a similar idea, but you'd be using grapes or fruit, and the sugars within that, the yeast would be feeding on. You could make sparkling wine. You have to be in France to call it champagne, so they say, because they're a little bit snooty, all right? If you wanted that, it would be the same idea, and you would have the alcohol that's been produced from your fruits. You would add a little bit of sugar back to it, bottle it and cap it, and it would carbonate within there, and it would be sparkling wine, all right? So all sorts of possibilities, and those yeast cells, the Saccharomyces cerevisiae, cerveza is beer in Spanish, all right? With the name like that, you ought to think we can use these for more than just making bread, right? There are numerous types or subspecies of Saccharomyces cerevisiae. Some of these yeast cells can tolerate low levels of alcohol production. We use those in making beer, and others can tolerate higher quantities or amounts of alcohol that are being produced. And we think about that with wine, right? Or potentially other spirits. And that stuff is basically the same idea like producing wine, but you're using something else like maybe corn. And once you have brought that concentration up to about 20% alcohol, then it gets distilled and concentrated to greater amounts, right? You didn't know you're getting a whole spiel about alcohol, but I blame you, right? You started, it's okay. So numerous possibilities, right? And I think it's really pretty interesting. There are some types of beer that claim they've got specific types of yeast that impart characteristics, and that's probably pretty accurate. And it really kind of just depends on those circumstances, right? As far as what they're doing and how well they're able to do it, all right? So fermentation gives you ethanol and CO2. If we are respiring aerobically, it's more just CO2 and the bread rising, right? And like I said, very low levels of alcohol that would be produced. This next image, I think it's interesting. It's also a bit disturbing as well as we look at a little of this and these open latrines. Very different than we think about today, right? You go into the restroom, you hope for a stall and a door, right? Unless you're at high school or middle schools. I think they took the doors off all of them because kids are just ripping them off, right? Ripping off the doors, ripping off the sinks. I'm like, what the hell is happening here, kids, right? I don't know. My kid came home from school. This was at the start, the very start of the semester. He's like, they just ripped the sinks off, Dad. I'm like, what? It better not be you, right? Okay. So I feel like they're trying to make us go back to this. You got to go to the bathroom, just go right beside your friend, right? That's a little bit disturbing to me, but these open latrines, that's how it used to be, all right? And it didn't really matter. You're not concerned about any of that. That's not really what we're getting at here. Although that's what disturbs me a bit. I want a little bit of privacy. We're looking at Rome and really their innovations. They had awesome aqueducts that were piping in fresh water into the city. And they also had mechanisms, sewage systems, like the reverse aqueduct, that will transport the waste away, all right? There are other issues, if we think about maybe like England, where they weren't following some of these same practices, and there's all sorts of nastiness, all right? We'll see a little bit of that later. I think it's impressive, all right? So the latrines, as we see here, and there's numerous ones, they would empty into the cloaca maxima. This is basically the huge sewer. And it, as we see here, running through town, taking the waste away, unfortunately dealt with by dumping it into the river, because that's how we dealt with most waste in the first place. And in some instances still do. That's kind of unfortunate. But the water through the aqueducts that they are bringing in, the fresh water, is not coming from the same place where the wastes are being dumped. And that is important when we think about it, all right? In England, as we think of like big cholera epidemics, that wasn't the case, all right? There was all sorts of issues, all right? Let's see, as I'm thinking about that, let me pause here. Any questions or concerns as I'm switching gears for one moment? All right, let me see if I can find this real quick. Any of you guys watch some Weird History?
No,
No? What the heck? Why not?
I didn't.
Need this. See if I can get it better. All right, this, I want to show you this real quick. I think this is crazy.
Customize the save.
And I need to, if I'm going to show you this, I've got to come back here. Got to share it before I can play it. Oh my gosh. When That looks. Oh man, and the great stink. That's a good one too. That's good stuff. So let me switch it back. The Broad Street pump and the map and all of that stuff. I mean, John Snow, we say the father of epidemiology. There's, as it ends, you know, getting rid of the miasma and moving into the germ theory. At the time when we're dealing with this and thinking about cholera, they had no clue what cholera was. They didn't know that it was a bacterium. They thought some sort of crazy poison or toxin, again, probably in the foul smelling air. That makes me back it up and think about that basement cesspool. Could you imagine how smelly everyone's house that has indoor plumbing, it just goes to the basement and there it sits, right? Gross and nasty. Some of it that actually got plumbed in to the sewage that's going to dump to the river would maybe alleviate some of that. But a lot of it, that's pretty disturbing and makes me think back to this, right? Maybe that was a little bit better. So Robert Koch, that we will see here in a second, he ultimately was the one that recognized Vibrio cholerae as a bacterium and the culprit for this disease. And if I remember right, it was about 20 years, give or take a little bit, after John Snow solidified this and got the pump taken out of commission. Pretty crazy. So with epidemiology, that I just said, John Snow, we say is kind of the father of epidemiology. He's the one that set the stage for asking these questions, looking and doing surveys, trying to figure out things and trace patterns that's still utilized today. It's pretty impressive, right? Okay. Enough of that. And I think there's maybe a couple more that are on there, the weird history that I'll try to share with you as we progress through. So a couple of historical tidbits as we move through here, and there were a few that I did get some Wikipedia links added in there if you are interested. I'm not going to click on that. Hippocrates, as I said, he had talked about the miasma. He had thoughts about natural and not supernatural causes, even if some of it might be the malodorous air. There are issues, all right? Malaria is named that because they thought it was bad air, foul-smelling air, malaria, that ultimately caused the disease. Big leads with the Athenian plague. I looked at that stuff. There were all sorts, 20 or more different potential culprits for the cause of this plague. I don't have an answer for what really caused it, but some sort of microbe for sure, right? And then as we get down to the bottom here, Varro saying that minute creatures, these microbes, that aren't able to be seen, they're what's potential culprits for disease, right? As we start moving forward, now we start getting some more details. So Pasteur, our good friend Louis Pasteur, he does all sorts of benefits to microbiology. Both of these two we really think of as fathers of microbiology that really kind of set the stage for understanding these microscopic organisms in the first place, all right? So Pasteur has all sorts of benefits to humanity. They say that he saved the silk industry with this type of, I believe it was a fungus that was decimating these moths. He dealt with that. His first bits with pasteurization had nothing to do with milk, all right? It had to do with beer, all right? And nobody wants their beer to get spoiled. What are we gonna do? Maybe if we pasteurized it, it would stay better, right? And then we shifted over and think about other stuff like milk, and pasteurized milk is a staple, right? Although you can find some sources of raw milk, I question why you would want to drink that in the first place. But we'll leave it at that, all right? So all sorts of advances in micro and immunology as well. The same with Koch. So studying the organisms, the patterns of disease, and the ways that our bodies potentially try to combat that were things that both of these individuals were really focused on. Robert Koch, I mentioned cholera a second ago. He studied numerous pathogens, anthrax, cholera, and TB. And we think, as we were talking or showed that video and they mentioned moving into this germ theory, this really goes to Koch. And as we think about his studying of these pathogens and trying to think about how disease is spread, he came up with a handful of postulates. We call them Koch's postulates. And it's not 100% of every disease-causing organism has gone through this, but the bulk of them have, all right? And the idea with this is, you see some sort of disease, all right? You are able to isolate some microbe, a bacterium, because that's what we're looking at here, anthrax. And he's looking at like cutaneous anthrax. So you get these lesions on your skin. He'd scrape some of that off of a patient that has the anthrax lesion and inoculate it onto some sort of culture media, like a potato slice, all right? We're gonna work with all sorts of Petri dishes that have all sorts of nutrients in there. This is super old school, right? We're just figuring out what's up. Slice a potato, rub some stuff on it, and it can grow, all right? There's lots of nutrients in that potato, right? And we will ship, there's a boatload of other stuff that Koch comes up with, or his grad students, right? So he grows it up. Now we've seen it on this patient. I grew it up on my media or my potato slice. Then I could take that and I could inoculate it into something else, maybe a mouse or, a lab animal, and inoculate it. And that the same pattern of disease shows up on this animal, right? That in turn was his idea to show this pattern of disease and really the idea with these postulates, right? There's another individual shortly after that put one extra step into Koch's postulates and basically said, now I've got my little mouse that's showing this lesion of anthrax. I can scrape that from the mouse, put it onto a new potato slice or a new Petri dish, and it grows up. And this plate looks the same as that plate. It's the same looking stuff. It's the same looking infection on both individual and lab animal or whatever it is. It's the same. This thing caused that disease, right? That's Koch's postulates. And like I said, the bulk of infectious disease we have verified with Koch's postulates and isolated it and showed this is truly what's happening, right? In the micro labs, when we start this stuff, we're kind of thinking similar sort of an idea. And we want to go through these processes to evaluate stuff. Really, as we think about the techniques in the lab, it's more of what we kind of describe as classical microbiology, right? We want to show you how the bacteria look under the microscope. We want to show you how they grow on specific Petri dishes, specific types of culture media. And we want to show you different types of biochemical tests, different types of sugars or other nutrients that we could supply to them that they may be able to grow on or maybe not. And based off of those possibilities and those reactions, we can pinpoint specific types of organisms, right? There are other techniques, and we think about, modern approaches utilizing DNA and sequencing techniques where we can extract DNA from an organism and figure out exactly what it is, its signature. There are techniques where you don't even need to cultivate stuff. You can just take a sample and sequence and understand more details, all right? But ultimately, having a foundation of how these organisms behave is very important, right? Not just scraping it and putting it into a machine and having it spit out an answer, right? Understanding what it is important, right? So both of these huge advances and really establishing our foundations of microbiology in the first place. Pretty cool, right? So this image, a couple of images here. Legionella it's not new, all right? But as far as infectious disease goes, it's pretty new on the scene, all right? It was first recognized and described as a pathogen in 1976, I believe. It was a convention, a Legionnaires' convention. So like, old Army veterans that basically American Legion, these are the Legionnaires, they got together for this convention and a good handful of them come down with some sort of crazy respiratory infection. And then we flip it back to thinking about Jon Snow and what are we going to do? How do we figure out? There's all sorts of epidemiology that went into play, trying to figure out what these guys were doing, what sorts of stuff they ate, where were they frequenting in this convention center. There was massive amounts of air conditioning units that were contaminated with this bacterium that they ultimately figured out that were spewing it out, blowing it into the rooms, and that's how these guys got infected. But it took a super long time to pinpoint that type of stuff back to the, House MD. Again, there's a couple of links on here that I put on there, but showing you that video, I don't want to link out to all of this stuff right now. So this is showing it fluorescing. We've got a black light that is shining on it that the bacterium fluoresces. There's handfuls of bacteria that will do that. It's not diagnostic of that, but one thing, and it's not the same plate, one thing that I do want to say, when they were first trying to figure out this bacterium, this culprit for disease, they couldn't cultivate it. you will see all sorts of different types of Petri dishes with all sorts of a rainbow of colors of media that's in there, all right? But you're only seeing a fraction of what's truly available, all right? And there are all sorts of other possibilities where some weird oddball organism needs something super special to grow. It won't grow unless you give it to it, all right? And it makes me stop and think, who's figuring that stuff out, right? Who is it that's doing this? That's like, oh, maybe a little of this, a little dash of pepper up in there will help this sucker grow, right? There's all sorts of crazy things that get added, amendments to media, right? The Legionella needed all sorts of specialized ingredients into these plates. Once this plate was formulated, it popped out, it started growing, and they were able to show this is what's up, Koch's postulates, right? Really pretty cool, right? This other image, and it's similar with this plate, you can see the bacteria spread all over, all right? The inoculating loop that we see over there is a tool that we will utilize to take bacteria from one thing and put it onto something else, onto a plate. Take it out of a test tube, maybe a liquid or a broth culture, and put it onto a solid media, right? As we look at this, and we're going to describe it in more details a little bit later as well, but this plate, what has happened on it is called streaking for isolation. And almost always in the lab, when you do stuff, you're going to try to do this same thing. And you take your bacteria and you spread it into the first bit on the plate. You take that loop and you will put it into the Bunsen burner, cook all of the bacteria that was on it so that it's dead. You let it cool off a bit and you come back to the plate and you spread some of it. And you cook your loop and you cool it and you spread it again. And you do that a couple of times. The idea is basically like creating a dilution or thinning out that bacteria on the plate. So you go from mass quantities to individual colonies, separate, distinct bits where a single bacterium has replicated countless times to give you a visible colony, all right? And that's what we hope to achieve when we're growing stuff up in the labs, is individual bits like this, whatever it might be. Okay, guys, we will quit with that. Have a good day.